recording and say welcome to Sonia Kéfi to give this IIT seminar. That will be the last one before we have a break for the summer, but we'll be back in the autumn, hopefully. And she's going to speak to us about the multiplexity of ecological communities. Thanks so much.
Well, thank you very much for the introduction and thank you for the invitation. And thanks to all of you for attending today. I'd like to start my talk with mentioning and acknowledging my colleagues and collaborators who are all part of the work that are present in this talk, and their names will come back throughout my talk. So in this talk, I'd like to raise the question of the importance of the diversity of interaction types between species. Ecological theory, as you well know, has been mainly based on a few interaction types, mostly predation, competition, but also other interaction types such as plant-pollinator interactions, for example. But if we look at all of this body of literature, we've clearly focused on a subset of interaction types compared to the great diversity of ways species influence and depend on each other in nature. So we may wonder if we've missed something, and whether we can actually improve our understanding, but also our predictive ability of natural communities by incorporating the diversity of interaction types which we know coexist in nature in ecological theory. So in this talk, I'll present a few studies that aim at contributing to answering that question. Well, ecological communities can be seen as complex networks of interdependencies. So on this figure, you can see here the disks are species and the white lines have feeding links between species of an intertidal community in Chile. And because communities can be seen as this complex network of interdependencies, it's knowingly very difficult to predict how perturbation that can happen in one corner of the web will propagate through the complex network of interactions to eventually affect the entire community. And this is, of course, becoming an increasing concern in the current context of global change and in particular of species extinctions. Well, the question of species extinction has been of interest for a very long time in ecology, both theoretically and empirically. And so I'll start my talk with an example of a study which highlighted a discrepancy between ecological theory and experimental work. So this is work that was led by my colleague Ian Donoghue, who's a professor at Trinity College in Dublin, Ireland. And so Ian and his colleagues, they performed experiments in the field in the rocky shores of the east coast of Ireland. And their purpose was to investigate the consequence of species loss from different trophic levels. So these communities, they can be simplified in such a cartoon network. We have groups of predators that are crabs and whelks that consume primary consumers that are grazers and mussels. And the grazers, they consume macroalgae that are composed of about 16 macroalgae taxa in this web. And so in this community, my colleagues simulated the local extinction of some of the species. So more specifically, they simulated the extinction of the crabs or the whelks in isolation or in combination with the extinction of the grazers or the mussels. And they let the experiment to run for 14 months. And at the end of the experiment, they quantified total biomass for each of the groups and secondary extinctions, which happened among the 16 macroalgae taxa. And so here is what they found. Here is the number of secondary extinctions when nothing happened, so that's the control, and when the whelks were removed or when the crabs were removed. And they found that the removal of either one of the two predator species led to 5 times more secondary extinctions than if nothing happened, than in the control. Interestingly, if now they remove the consumers, either in isolation or in combination with the predators, they don't find that result. So in that case, there is no significant increase in secondary extinctions. So basically what they found is that the removal of the predators and the predator alone led to about a third, the extinction of about a third of the microalgae taxa. The removal of the predator alone led to a very large number of secondary extinction. And this is interesting in many ways, and actually the study goes into the mechanisms by which this happens. But one of the interesting conclusions of that study is that actually this number of secondary extinction is much larger than what most theoretical studies predict. Actually, it's an order of magnitude larger. In most theoretical models, actually, secondary extinctions occur rarely following the loss of predators, but they're much more likely when we remove species that are either at intermediate or basal trophic levels. So these experimental results, they're much greater than the theoretical models, but they're in agreement with traditional classical studies in ecology, including the work of Robert Payne, and his famous experiment he performed in the 60s on the rocky intertidal zone in Washington state. And so Robert Payne worked on these communities that included 15 species initially. And when he removed the starfish and followed the community through time, this led to a large number of secondary extinction, and eventually the community ended up with a single species, the mussel. So this is a classical example of how the removal of a single species can destabilize an entire community and lead to many secondary extinction. So there seems to be a puzzling discrepancy between these observations and the predictions of theoretical models. So what are the missing ingredients in our models? Well, actually, these models that I've been referring to in a very generic way, they're food web models. So they are models that model feeding interactions between species. But if we go back to a picture of an intertidal community, this is a picture of the intertidal zone in Chile. These communities, they show great diversity of interaction types. So the feeding interactions are very well studied in these communities. Here you see heliasters searching for food, or you see chitons that are herbivorous species. But you see other interaction types as well that are very well known and studied in these intertidal communities. So for example, here you see competition for space between algae. Here you see gelidium, this algae that facilitates the recruitment of mussels on the shore. And here you see three-dimensional structures created by mussels in which crabs can hide. And actually in these communities, food is not really limiting for crabs, but they do fight very fiercely for those hideouts where they're protected from waste, but also from their predators. So clearly, when you look at these communities, the species form very complex network of interdependencies where species depend on each other in many different ways. So going back to the network of the rocky shores of the east coast of Ireland, looking at it more closely revealed also very well-known non-feeding interactions. Mussels compete for space with macroalgae. The different macroalgae taxa also compete for space. And crabs and whelks are involved in what we call foraging modulation. This means that they both eat grazers and mussels. But actually, when they're alone, they prefer the grazers. So when they're alone, each of them prefer grazers, but when they're together, they kind of change their eating habit and they start eating mussels as well. So we're wondering how including these two types of non-trophic interactions would affect the prediction of a theoretical model. So we worked with a bioenergetic consumer resource model. that to simulate the dynamics of the species. So it's based on a model that is very well used in the literature, originally developed by Jossis and Ines, and then used a lot by Ulrich Broos and his teams. And the idea is that if Bi is the biomass of species I, its dynamic can be described with this general equation where you have a growth term for plants, then biomass gained by consumption for species that are not plants, a term of consumption by predators, and a loss term due to metabolism. And so in this equation, the Fij here is the functional response, which is here assumed to be a multiple holding type functional response. So again, this is a model that is quite widely used in the literature. So we start with the food web that is observed in the field. So this is what determines the structure of the network. And we're going to map this equation to that food web. But now what we want to do is incorporate the non-trophic interactions. So the two groups of non-trophic interactions that have been identified as important for this community, competition for space and foraging modulation. In the bioenergy, in the food web model, we use allometric scaling to kind of parameterize the model in terms of everything that is related to feeding. So we have a kind of generic way of parameterizing the model for the feeding parameters, but we have no idea about the intensity of the non-trophic interactions. So what we're going to do is we're going to run simulations for a range of parameter values for the intensity of competition for space and foraging modulation. And this are simulations that were run by a previous student, Alexandre Génin. So here, in this figure, you'll see outcome of the simulation for an increasing strength of foraging modulation on the x-axis and increasing competition strength on the y-axis. And so what Alexandre did is that he replicated exactly what happened in the experiments, in the field experiments. So he starts with a network as I showed you earlier. So all the species are present. He runs a simulation into steady state, and then he removes the predator one by one, either alone or in combination with the removal of a primary consumer. And then he checked what happens in terms of biomass and secondary extinctions in the model. And then this first figure here, you'll see the parameter space corresponding to the cases where the simulation results match the experiment in terms of number of extinction here in green, and in orange where it doesn't match the experiment in terms of number of extinction. And so here is what he got. What is interesting, firstly, is that here in that bottom corner is where both non-trophic interactions are zero. So this is a food web. So in the case of your pure food web, without any non-trophic interaction, what you see is that there is no secondary extinction. So in this model, we cannot reproduce the result of the experiment without any non-trophic interaction. And you see that there is here in green an area where the model is able to reproduce the same amount of secondary extinction as in the experiment, and that happens for intermediate competition strength. Now, I mentioned that in the experiment that did not only follow extinctions, but also biomass trends. And what happened is that when either one of the predator was removed, there was a large number of secondary extinction, but there was also an increase in the biomass of the mussels by 20 to 60%, while the whelks didn't change their biomass. Oh, sorry, the grazers. And so in the second figure, in yellow, you'll see an area where the simulation matched the result of the experiment in terms of trend of biomass after the removal of the predator, and in blue where it does not match the experiment. And what you see here is that the simulation can reproduce the experimental result for intermediate competition strength and for intermediate to high strengths of foraging modulation. So this is an example of a model where the diversity of interaction types, so both interaction types simultaneously, are needed to reproduce the results of the experiment. So it's one example of a way of resolving this discrepancy between the observations and model predictions. There might be others, but it's a way this could be resolved. But these results are for one specific case, for one field experiment. And based on this first study, we were wondering, more generally, how does the diversity of interaction types affect species coexistence and the functioning of ecological community? So this is work that was done with my colleague Vincent Miel. And we started with the same bioenergetic consumer resource model. And now we incorporated a broader range of non-trophic interactions: competition for space, interference between predators. So when predators have prey in common, they might fight for accessing those prey. Recruitment facilitation. So in the examples I gave earlier, Gelidium, the algae, was facilitating the recruitment of mussel on the shore. Refuge provisioning, that would be, for example, the mussels providing a refuge to the crabs that are then protected from their predators. And we also included positive and negative effects on survival. And so we build network food webs of 100 species, including 20 plants, using the niche model. So just to get a structure that is kind of realistic. And then in this model, we're going to plug in non-trophic interactions randomly, but with some constraints. So for example, competition for space can only happen between species that are sessile. Interference between predators can only happen between two predators that have at least one prey in common. So there's a few constraints, but otherwise the location of the non-trophic links is chosen randomly. And then we're going to run dynamical simulation with and without those non-trophic interactions. As mentioned earlier, we can use anometric scaling and the literature to try to parameterize, at least get a set of more or less realistic values for the food web part of the model. But for the non-trophic interactions, we have no idea of realistic values. And in particular, we have no idea of relative values. So how strong should competition for space be compared to refuge provisioning, for example? At least I have no idea. So we had to find a way of defining a range of values that made sense or that helped us decrease the enormous range of possible. And in the previous study, we had only 2 non-trophic interactions, so we could kind of run simulations around those 2 axes, varying both interaction strength simultaneously, but now we have 6 of them, and so that becomes more difficult. So this is what we decided to do. So again, we run each simulations by pair with and without the non-trophic interactions, so food web and a food web plus other types of interactions. And at the end, we measure variables, including how many species survive. And so, for example, we measure variation in diversity. So that's the relative change in species number due to the presence of the non-trophic interaction. So we measure how many species at the end of the simulation in the network with non-trophic interaction compared to the food web. And so if that term is positive, it means the presence of the non-trophic interaction in the web increase species diversity compared to a food web alone, and conversely. And we can measure this for different intensity of the non-trophic interaction. So as an example here, for interference between predator, here on the x-axis, interference between predator increases, and here is the variation in species diversity. So when it's 0, it means the incorporation of non-trophic interaction has no effect on species diversity. So we can plot this term as a function of the intensity of the non-trophic interaction. And in this case, for example, we get this. So as interference increases, we get less and less species in the network with non-trophic interaction compared to the food web. And we use this to calibrate the intensity of the interaction that we'll use by deciding, and this is completely arbitrary, that we'll take the lower intensity or strength of the interaction corresponding to a change of 2.5% in this term, in this ratio, and the maximum value to 10%. So this allows us to define a range that is now, we'll repeat that for all the interaction types. So then we'll have the same range for all interaction types. This means that by doing that, we're trying to put all non-trophic interaction kind of on the same scale. They all have strength that correspond to something that leads to between 2.5 and 10% change in species diversity compared to a food web. Again, this can be discussed, but this is what we did in this study. And so again, we put the non-trophic links randomly. We take a value for the non-trophic interaction in that predefined range that kind of tries to put them into a similar scale. And then we decided to put exactly the same number of positive and negative non-trophic links in all the networks. At the end of the simulation, we calculate species diversity and total biomass. And so, for example, here is how biomass changes. So the total biomass in the network at the end of the simulation changes as a function of species diversity, species number. In the case of a food web, so no non-trophic interaction. So in a food web, we find this typical relationship. Here's just an example, but you have an increasing relationship between species diversity and biomass. Now, if we include non-trophic interactions, this is what you get. So this is the red curve now. What you see is that the slope is steeper in the presence of non-trophic interaction than in a food web. And this is something that seems to be very robust and happen kind of consistently across different simulation runs for different parameter values and so on. And actually, it seems to be something that emerges kind of mechanically. So I'll try to explain what happens. If you look at those communities, remember that we take all the interaction strength randomly within that predefined range. And so these communities here that have a really relatively high species diversity, they tend to have a disproportionate higher biomass compared to the food web. And by chance, they're communities in which the positive non-trophic interaction are relatively strong and the negative non-trophic interactions are relatively weak. So as soon as you have very strong positive non-trophic interactions in communities, it will tend to increase species diversity, and this will be accompanied by relatively higher biomass than you would expect in a food web. Conversely, these communities here, they're communities where you have a few strong negative non-trophic interactions, and that decrease species diversity and the associated biomass even more compared to a food web alone. So what this suggests is that as soon as you have non-trophic interaction, and you have communities where you have a bit more positive and other communities where you have a bit more negative non-trophic interactions, it will probably make this slope steeper than you would expect if you would take only feeding interactions into account. Interestingly, this means that if you look at this curve to try to see what would happen in terms of a species extinction, something that is often done in the literature, this means that in a community with non-trophic interaction, the consequence of species loss for functioning here, biomass, is stronger than what you would expect by modeling the community by feeding interactions alone. So here in this second study, we found that incorporating non-trophic interactions affected species coexistence, total biomass, but also the relationship between species diversity and community functioning. So until now, I mean, this study the non-trophic interaction were plugged randomly. And this is because we didn't really know better. But one burning question we had was, what about their structure? Do we know anything about the structure of this interaction that could help us constrain the simulations? So how do different interaction types map onto each other? And actually, there's been a number of really nice studies that date already quite back. So Carlos Melian and colleague published in 2009 a network including 2 interaction types. You have plant pollinator, plant seed dispersal interaction on the one hand, and plant herbivore interaction on the other hand. And a few years later, Michael Pocock and colleagues published a network including 7 different bi- and tripartite network for an agroecosystem in the UK. And these studies, they showed that actually the structure of these networks were not random, and these tend to have implication for, in particular, secondary extinctions, the way perturbations spread in those networks. But so these type of representation, these multipartite networks, they don't capture the multi-trophic structure of ecological communities. So we got interested in trying to have data that would give us information about all possible interactions that exist between a given set of species. So what we call a multiplex ecological network. And so this is what we tried to do with my colleagues Sergio Navarrete, Ivy Viters, and Eric Bello. We compiled a full ecological network for the coastal marine ecosystem of Chile. And so the data set includes all species found to co-occur during community surveys carried out at different locations along about 700 kilometers of the central coast of Chile. And the network construction is based on the very rich literature available for these communities, a lot of experimental work, and the great expertise of my colleagues Sergio Navarrete and Ivy Viters, who have been working on these communities for decades. So the network includes 106 species. Here they're sorted out vertically by trophic level, with basal species here and predators here. And they're randomly spread along the x-axis. And the network include about 1,500 feeding links, more than 3,000 negative non-trophic links, and a bit more than 150 positive non-trophic interactions. So with this data, there's a lot we can do, but one of the questions we had was, are there patterns in the way species manage being involved in different interaction types? In other terms, can we collapse the species into a smaller set of multiplex clusters, meaning groups of species that share similar types of links with similar species? And so to address that question, my colleague Vincent Miel developed a stochastic block model approach applied on this network. And the idea is to group species based on their connectivity pattern across these 3 dimensions: the trophic, negative non-trophic, positive non-trophic. I'll try to give you an intuition of what that algorithm does. So the idea is, imagine that the nodes here are species and the links are feeding links. One thing that we can do is group species, but how similar they are in terms of who eats them and what they eat. So in this very simple example, you would have 2 big groups of species, what we call trophic species, because those 3 species, they have the same consumer and they consume the same prey. Now, if we add an additional interaction type here, for example, facilitation, we can define different groups. And here, for example, there is a group of 2 species that are similar in terms of the feeding links and the facilitative links. Now, if I add again another interaction type here, competition, for example, I can define another group of species that are similar in terms of feeding and competition. So that's the idea. We're trying to group species in terms of their similarity in the way they interact in the 3 different layers of interaction, as well as the partner they interact with. So is there some kind of three-dimensional connectivity pattern that is typical in this network? So starting from the 106 species and applying the stochastic block model approach, we got 14 clusters of species. So this is not a very informative picture. These are the clusters. Their size is proportional to the number of species in the clusters. But if I now go back to the species level, where here each disk is a species and they're clustered in space by the group they belong to, the cluster they belong to. And the colors here is their relative involvement into the different interaction types. So in blue is feeding, in red is facilitative interactions, and in gray is competition. What you see visually is that the algorithm does a pretty good job at putting together species that are similar in terms of how much they interact in the three different layers of interaction. So here, for example, you have a group of species that are mainly involved in competition, and they're eaten, basically the groups of algae. This is a group of species that is a multiplex cluster, so hub actually of species that are very much involved in the three types of interactions with many species in the web. But I won't go much more in detail. What the question we had after getting these results was, is there some kind of functional consequence of this multidimensional connectivity pattern? Does it matter that there's 14 ways in which species are interacting with other species along these three dimensions of interaction in the Chilean web? So to address that question, we use again a bioenergetic consumer resource model, in which we incorporate the 6 same non-trophic interactions that are actually the most common non-trophic interactions found in the Chilean web. And we're going to run numerical simulations of not the 106 species, but the 14 nodes. The idea is that in each cluster, go back, in each cluster here, species are very similar in terms of their connectivity pattern and in terms of who they interact with. This means that species that belong to the same cluster, so here those 2 species or here this group of species, they're very redundant. So instead of modeling the dynamics of each of the species, we're now going to model the dynamic of a typical species of a cluster by averaging its property. So we start with 14 nodes that are a typical species of a cluster. And we're going to run simulation where the connectivity of the Chilean web is maintained, and in which the non-trophic layers are randomized. So for each simulation, we're going to keep the food web layer constant, and we're going to randomize the facilitative, the positive non-trophic, and the negative non-trophic layer by keeping the degree sequence identical. This means that in the randomized networks, the species or the nodes are involved in the same extent in the different interaction types, but not to the same partners. So it's a very constraining kind of null model. And at the end, we measure how many species survive, how many nodes survive, and what's the total biomass. So here, what you see is that for the Chilean web, when we keep the structure of the Chilean web constant, you find that amount of biomass and the 14 clusters survive. But in the 500 random web, we tend to have slightly lower biomass and lower persistence, since a few of the nodes go extinct. So in this very constraining randomized web, what this suggests is that there is a very specific 3-dimensional signatures of the clusters, which tend to lead to higher species persistence and high total biomass. So of course, this is only one data set, the cheetah and web, and because there's very few data set of this type, this multiplex data set, it's actually very difficult to know to what extent these patterns are very unique to either intertidal areas or marine ecosystems, or can be generalizable to other type of ecosystems. And so this is the type of question we can only answer when more data will become available. But so with these few studies, what I tried to show is that by incorporating the diversity of interaction types in food web, so by studying multiplex ecological networks, we could in some cases resolve discrepancies, gaps between theoretical predictions and observations, that non-trophic interactions could increase the number of secondary extinctions following a primary extinction. That means that ignoring them would lead to underestimating the number of secondary extinction. It could affect coexistence, total biomass, but also strengthen the relationship between species diversity and total biomass. And also these non-trophic interactions, they seem to have a structure, and a structure relative to each other that could very well matter for species coexistence and the functioning of ecological communities. So all of these studies taken together, they seem only a few pieces, they seem to be only a few pieces of a puzzle, but they suggest that going towards that direction of trying to figure out when and why the diversity of interaction types matters may be a worthwhile effort to improve our understanding, fundamental understanding of ecological communities, and eventually our predictive ability. So in this talk, I focused on multi-interaction networks. But I'd just like to hint at the end of this talk that actually it's not the end of the story, because there is increasing work on networks that include different snapshots in time or different snapshots in space, and there is an increasing number of tools to analyze such networks, the multi-layer framework, and this could also extend the range of questions that we can ask even further. And with this, I'd like to thank you very much for your attention, and I'd like to acknowledge my collaborators again on the work I presented to you today.
Thank you very much. Thanks a lot. If anybody have any questions, best way to do that is in the reactions bottom. Just click the raise hand, and we can unmute and ask. But yeah, thank you again, Sonia.
Thank you.
So I think there's clap emoji pictures which are different to the hands up. So we'll see. If no one's got any questions right now, I'll ask one thing which I was wondering. So why don't you think other people have tried to create these non-trophic interaction networks too? Because I guess it's been, well, 7 years now since you put the Chilean one out. Is it just a case of people not having the money to know to do it, or do you think, I mean, because one good network exists, it's like, oh, we can just use that one? Or
yeah, that's a very good question. I have to say I've been a bit disappointed by that. I thought this would initiate more enthusiasm in the community. I guess there's probably different reasons, but one of them is that there's probably, I don't know if the few studies that are available so far are convincing enough that it's a worthwhile effort. So what I hear a lot is people consider this diversity of interaction types often as noise. And I guess it depends what kind of communities you're studying, but it's probably not in all communities that the diversity of interaction types are all important. And so I think for me, one of the big questions is, when do the diversity of interaction type matters? What interactions matter? And actually, to understand that, to answer that question, we need both theory and data. And data is currently a huge limitation, because in models, I mean, you can do a lot, of course, but the dimensions, the number of dimensions is enormous. So we need to find a way of constraining things. And yeah, so I guess part of my answer is that I don't know to what extent people are convinced that it is an important question, at least that it could improve our understanding and or predictive ability. And the second part of the question, the second part of the answer is that it's actually very time consuming to do. Yeah, you need to dig the literature, you need to have experts that know the community very well. And even when it's the case, so I think the intertidal communities have an enormous amount of knowledge for a long time on diverse interaction types, but it's still, I mean, it's a huge amount of work to gather all these interactions. And then still, there's enormous biases, right? Because we've had interest in some interactions and not in others. So yeah, what we have, what we can collect is only the reflect of what we as ecologists have been interested in. And so this also makes it difficult to sometimes maybe to justify the effort in gathering this amount of data. Yeah, that's the reason I see. If other people see other reasons, I'm happy to hear what people think. Yeah.
No, I think I agree. I mean, but sometimes from the simulation angle, you have the same problem because it hasn't been demonstrated empirically that you kind of need to do these things, and then people don't want to add in everything. It's especially when it can very easily send your model off to infinity if you build in the wrong or sort of facilitation. So then you have to make assumption, and before you know it, then you're not really doing generic modeling anymore. But so yeah, Hugo, yeah. Do you want to ask?
Yeah, I just had a question about, like, how do you, yeah, about your non-trophic interactions, and you said that it's like the difficulty in kind of, like how do you determine like how strong those interactions are? So that's kind of my question is like how did you go about like deciding how strong each individual interaction would be?
So in the BEF study, right, where we put them all between 2.5 and 10%. That's your question, or I guess.
Yeah. Wait, could you go back to that slide?
Sure. Yes. Let me check. Yeah. So hop. Wait here. Oh, this one?
Maybe. Like, yeah. Yeah, it might be what I'm asking. I'm not sure. But yeah, I was just thinking, like, you had your list of like non-trophic
yes
interactions, and like I was just thinking, like, some of them are like quite different to each other. So like I would, how do you how do you determine kind of how to model those interactions so that they have an effect on the dynamics?
Yeah, so that's a very good question. We don't really know. And so if you only focus on a few interaction types, like in the first study, what we did was to just run simulation for all possible combination of parameter values, and then we see what are the different behaviors of the model. But in this study, for example, where we had many different non-trophic interactions, we tried to find a way of putting them on a similar scale, which may not be realistic because, I mean, I don't know, maybe competition for space is actually much stronger than refuge provisioning in nature, but we basically don't know. There might be some resources, but I don't really know in general if there's any information on relative strengths of different interaction types. So what we did was we decided that we wanted to put them on a similar scale in terms of the effect on coexistence. So we run simulations with different intensity of each of the non-trophic interactions. So here it's interference, and we define the range of values that we're going to use as an effect of 2.5 versus 10% on species diversity. And we do that for each non-trophic interaction. This means that in this case, for interference, it's 0.75 to 3, but for other interaction types, it might be 10 to 100. You know, so then you have completely different values for each of the non-trophic interaction, but you know that they have that limited effect on species diversity. And so that means that when you put them together, at least that was our expectation, we put them on, we fix the range, and then we put the same number of positive and non-trophic and negative non-trophic interactions. So we expect the effect on species diversity to kind of cancel each other out when you put them together. And what we see is actually this. So in terms of species diversity, they should kind of cancel each other out. But what we find is that the effect on biomass is disproportional. So when you have non-trophic interaction that lead to high species diversity, it increases biomass disproportionately compared to what you would expect in the food web alone. So it's in this particular case, because we were going to plot biomass as a function of species diversity, it seemed, I don't know, one option to kind of put all the interaction types on a similar scale based on the effect on diversity. But of course, we could have decided something else, right? It's
no, but it sounds like a reasonable approach, right? You kind of set the parameter values so that the outcome, the effect on your diversity is kind of in this was it 2 to 10%. 10%, yeah. Okay, yeah, thanks.
But in nature it might be completely different, and then
yeah, sure. But I mean, like if one interaction is super small, then it's just you're not going to, and you want to look at the effects, right? So yeah, that makes sense. Yeah, thanks.
Thank you.
I don't see any hands up, but if anyone wants just to unmute and ask, if you can't find, sometimes the button, people don't find it, but if there is any questions.
But in the meantime, I can just add something to what you were saying. Actually, it's always a question. I think as a theoretician, we want to make models that are simple, but then you always realize there's missing dimensions, like missing species diversity or traits, and it's always a question of which are the directions of complexity that are important for whatever you're trying to understand or predict. And so the question of whether and when interaction diversity is one of these essential dimensions remains largely open. So again, like here, we did a few studies in which we try to show in specific cases why and when it seems to matter. But I think what we lack is a kind of general framework that tells us when and why this diversity would matter, and that would help tremendously in terms of both data collection and building our understanding.
So even within the kind of, all these are kind of direct, I guess, pairwise interactions, and then if you step up to the sort of higher order interactions of all their various flavors and how to describe them, because many of these non-trophic interactions, they're caused by interaction modifications of some sort.
Yeah.
Whether it's worth stepping up to that level, I think is another, because in some ways it simplifies things, because but it also complicates in other ways. But
so actually several of these interactions are already higher order interaction, because so for example, predator refuge provisioning, for example, so a species protects another from all its predators. So it's already an interaction that involves more than a pair of species. We have a bunch of modification of feeding interactions. So there is no higher order interactions in those non-trophic interactions that are involved here. But it's again, another question, how the higher order interactions specifically affect the results compared to interactions that are pairwise. And I don't know the answer to that.
Looking at it, seems more like the multi-layer networks have almost overtaken the higher order as sort of the new thing to look at in many ways. But they're different in a way that's they're different enough, but at the same time, they sort of try and capture similar things. And it, I don't really know what the answer is, because there's been so many papers talking about different definitions of these things, but
Yes.
we need data somehow, and that's it's really difficult. I'll do. Were there any final questions from anybody else before I'll stop the recording? There's lots of people still in the audience, which I guess means they're waiting for other people to ask questions. Maybe they're all heading off now. Okay.